In this video, we're going to take a look at transcription, which is essentially the first process involved in protein synthesis. If you're not sure what mRNA, tRNA is, or nucleotides, then there is a range of videos that you can watch, at least on this channel, just to make sure that you know what they are before you get started with this. The first thing that happens is an enzyme attaches to the DNA. The enzyme is called RNA polymerase, and that attaches to the DNA at the beginning of a gene sequence. It has the effect of breaking the hydrogen bonds between the two DNA strands, between the bases, and that causes the strands to separate and exposes the bases of the DNA molecule, as you can see in the diagram. One of those strands is then used to make a template, is then used as a template to make an mRNA copy. In order for this to happen, the RNA polymerase essentially lines up, attaches RNA nucleotides along the exposed bases in the template strand. Now, these RNA nucleotides, don't panic too much about where they come from. They are found in the nucleus as free RNA nucleotides. When they find the bases that they can associate with, they will combine and they will begin to form a polynucleotide, which is the mRNA. The key difference with that, as we mentioned before, is it would be exactly the same as the DNA base pairing rules as a complementary strand. However, they don't have a thymine base. They only have a uracil base. So where you would find T, you will find U instead. Once they've paired up with their specific bases on that strand, they're joined together by RNA polymerase, which then makes this mRNA strand. The RNA polymerase then moves down the DNA strand, so it continues to move along the DNA, and as it does that, the mRNA strand continues to be assembled. The same process occurs. The hydrogen bonds between the uncoiled strands break apart. It means that the RNA nucleotides can fit in, RNA polymerase attaches them, and then afterwards the DNA reforms once it passes. So it's a little bit like the replication bubbles. It's a small section where the nucleotides are being assembled. This leaves a trailing mRNA strand that is being formed as the RNA polymerase moves along. Once the RNA polymerase reaches a stop signal, and that's a particular sequence of DNA called a stop signal, you can take a look at those in the other video which I did on mRNA. Once it reaches that, it will fall away, and then the mRNA can move out of the nucleus through a nuclear pore, and it associates with a ribosome in the cytoplasm, and that is where the next stage of the protein synthesis will take place. One other factor that needs to occur with the mRNA, the molecule that we've just described being made, is called pre-mRNA. Now, in eukaryotes, we have pre-mRNA. In prokaryotes, they don't. So let's focus on eukaryotes first of all. There are sequences called exons and introns. An exon is a sequence that is used in coding for a protein. Introns are called non-coding, so they don't code for the protein and they need to be removed. A process called splicing occurs, which essentially means that the exons join together and it pinches off the intron, and you're left with just the sequence of exons and not introns. It can be tricky to remember which is which, exons or introns. Sometimes I feel like I kind of would want to remember it as exons are the ones that get removed, but it's the introns that are taken out. That then makes the mRNA strand. So the sequence that we had copied directly from the DNA is called pre-mRNA, but once the splicing has removed those introns from it to just the coding, just the bases that code for the amino acid, that is called the mRNA strand, and that is the one that goes on to the process of translation. In prokaryotes, there are no introns, so it doesn't need to go through the splicing process.